So we had the opportunity to look at the whole exome sequencing landscape of appendiceal adenocarcinoma. And so this is a descriptive study of over 800 patients with appendiceal adenocarcinoma of all stages who were sequenced as part of their Natera Signatera test that develops bespoke probes using whole exome sequencing. And so collaborating with Natera, we were able to examine the whole exome sequences of these tumors in order to quantify the largest series to date of appendiceal adenocarcinoma whole exomes. So this is simply an observational design, observational cohort study, looking at again, just the genetic sequencing of these tumors. We were able to recapitulate previous associations with that demonstrated that the tumor mutational burden and microsatellite instability status of appendiceal adenocarcinoma was distinct from matched cohorts of rectal and colon cancers, and that especially the mutational burden was significantly lower in appendiceal adenocarcinoma. And then when we looked at the molecular subtyping of these tumors, we found that they, as previously described, follow subtyping categorization as KRAS-like, GNAS-like, TP53-like, and triple negative, and that there were distinct profiles associated in copy number variation as well as ctDNA status and stage were associated with this subtyping mutational status. And again, we were able to demonstrate that the genetic landscape of appendiceal adenocarcinoma is quite distinct from both colon and rectal cancers, with a much higher prevalence of GNAS mutations and a significantly lower prevalence of APC mutations, as well as TP53 mutations. Yes, that's a great question. We are currently looking at a cohort within MD Anderson with more robust clinical data so we can better understand how to take these whole exome observations and apply them clinically for our patients. While we see that there appear to be more aggressive subtypes of appendiceal adenocarcinoma, um, such as the TP53 subtype, how that actually relates to patient care is yet to be further defined. And so we're in the process of applying that to our patient cohort here at MD Anderson. And then similarly, when we looked at the mutational signatures, while tumor mutational burden and MSI status were typically low, we found that the mutational signatures carried a mismatch repair deficient subtype in a significant proportion, up to 20%. And so there are ongoing studies at MD Anderson and elsewhere looking at the role of immunotherapy and checkpoint blockade in these patients, and if could potentially be a benefit to that subset of patients with appendiceal adenocarcinoma.